Hi everyone, today we'll be talking about triplet codes. Triplet codes are also known as codon, and from the name itself, triple meaning three, it is a code made up of three letters which are corresponding for an amino acid. Triplet codes are always read at the same direction, 5' to 3', and the complementary strand is known as an antisense strand of the DNA. This does not carry the translatable code in the 5' to 3' direction. The sense strand is a strand of DNA that has the same sequence as the mRNA, which will later on be discussed in protein synthesis. How do we read triplet codes? In order to read a triplet code, you would need a copy of the table found in page 513, 513, Appendix 2 of your textbook. This one shows you a table that has the first position, second position, and third position. And in the middle, there are codes for amino acids. In order to see what amino acid possess or what code does the amino acid have, you could refer to page 512. In the video, I'm only showing a general picture of the position on how to read these codes. The dark blue shaded area will be the first letter. The one shaded with orange will be followed by the second letter in the code, and the last one, the column to your right, which is lighter blue, that will be the third letter of the code. When you check the table, you would actually notice that one amino acid have a lot of codes for it. That is fine, that is the usual, but you would have to be careful in reading it and matching it in order for you to not create a mistake in reading which code is which. Let us take CTA as our triplet code for an example. So let's pull up the image of the table in here. Then locate letter C. I will be enclosing that one in a green box. This means that our amino acid is most probably in this row. Next one, locate letter T, the second letter in the second position, and we'll use color blue. It's there. Let me box that one. Our amino acid most likely is in this column. And lastly, look for letter A, which is the third letter in the third position, the last column to your right. Let's use yellow, and it is letter A, right? Let's encircle that one and pinpoint it. There you go. See, A is an amino acid known as aspargine. 15 seconds, look for the amino acid for ATG. As always, first letter, look for it in first position. Let's encircle A. Next, let's look for letter T in second position. And then, lastly, the third letter in the third position, which is G. And that leads us to the amino acid tyrosine. Next example, let's try CGC. You can pause the video while you're looking for the image, but I'll give you 15 seconds. If you got ALA or alanine as your answer, therefore you are correct. Now that you have explored the codon table, you might have noticed that there are some that are not having or have a weird word in it like stop or start. And this means that the codons are actually separated into different groups or different types. We have three types of this. First one, we have the normal one, which is the one that has an amino acid symbol beside it. Then we have our start codon, which is symbolized by a UG. This one is also known as methionine. Okay, this is the codon that will start the protein synthesis in the ribosome. But do note that if AUG, the codon AUG, is found in the middle of the whole length of code to be translated by the ribosome, this will not register as start, but rather it will be read as methionine. And lastly, we have three special codon to indicate the stop of protein synthesis, and that is UAA, UAG, and UGA. These three do not code for any other amino acid other than the indicating the stop of or the end of protein synthesis in the ribosome. Once this one is read by the ribosome, the polypeptide chain that is produced will now be released and will now go to folding stage in order to reach the secondary, tertiary, and quaternary structure of a protein. We said before that the cell cycle have a lot of checkpoints in different section or phases of the cell cycle, but there are times where a mutation can still persist and still occur. And an example of a mutation that is commonly found in humans would be sickle cell anemia. Sickle cell anemia occurs when a mutation in the gene coding for hemoglobin has a mutation. Rather than having the usual biconcave shape, the beta sheet is affected and there are codes that are swapped with each other or are substituted with a different amino acid. Let us represent the gene or the nucleotide sequence that codes for a protein with the letters HB. But for the normal one, we will use the code HBA. And in the sickle cell red blood cell, we will be using the code HBS. Let me pull up an image in here. Okay, so in here you could see that the gray area, the glutamic acid, has been changed into valine. But the change in the code is not even as drastic as the whole three codes changing or missing, but rather there's only one letter in that triplet code which is substituted into another one. And in this case, it affected what type of amino acid will be attached to the new hemoglobin. This type of mutation is known as substitution. So that'll be it for this video. See you in the next one. Don't forget to answer the worksheet. Bye!